So I'm very pleased to welcome Monica Olivera de la Cruz. I wish I could pronounce it correctly, but I try. It's actually quite a delight to have Monica here. She's one of our colleagues from close by at Northwestern. She's been on the faculty there since 1986, so essentially your entire career as a faculty member, rising to her current position as the Lawyer Taylor Professor of Material Science and Engineering, and she also holds professorships in chemistry, physics, and astronomy, and chemical sciences and engineering. Among her many honors, she is a member of the National Academy of Sciences, AAAS. She was a recipient of the Kazaroli Prize of the NAS. She has received a Presidential Young Investigator Award, a Sloan Fellowship, a Packard Fellowship, and actually in many ways most important from the standpoint of a Department of Energy facility funded by Basic Energy Sciences. She is a member of the Basic Energy Sciences Advisory Committee, so very instrumental in helping BES steer the course which we try to follow. Her research interests include elastic shells, electrostatic assembly, electrostatics at liquid interfaces, ionic gels, and co-assembly, and the behavior of complex molecular fluids. And I think largely speaking, her research is focused on theory and simulation of increasingly complex systems. And it's, again, a delight to welcome her here, and I think we'll hear about your work on polyhedral elastic shells today. So thank you for joining us.
Thank you so much for the invitation. I'm having a great time. Every time I visit here, I'm so fascinated with the science, and I learn a lot. So I'm going to try to tell you a work that we have been doing on this topic that involves people, collaborations that we have with other groups. So this is members of my group, the theory and simulations from Rasco, Creighton, and Bafu, which Bafu will be joining here next year, the group in Linda and with Ellis Ross. And I work a lot with Sam Stoop, and this is a student that we have before co-supervising. And also with Mike Betsick, which uses a lot of the facility here. And this student was working the project also co-advised between us. So let me just go through an introduction of polyhedral. So all of you are familiar with the Platonic solids, which had, these very beautiful geometrical features, like you can characterize everything about them, knowing like the rotations and all the symmetries they have, by knowing how many lines are in each vertex and the number of sides of the polygons that made them. They are all the same. They are made of the same single polygon. And so you have these 5 Platonic solids. And they have been fascinating scientists. They also fascinate artists and many groups. But the scientists have been particularly interested in Platonic solids because they are so geometrical. So here's an example of Plato, who had assigned for each element one of his Platonic solids, right? So the fire, and wind and sea and earth. And he needed one more, so he said the cosmos should be the dodecahedra. So the dodecahedra, and I was invited to give this talk on polyhedra in Wisconsin. And apparently, in a group in Wisconsin in 2003, they have discovered that the radiation of the Big Bang had a symmetry of the dodecahedra. So I always think this is a very fascinating story because, the ether, Aristotle came and said there's ether, and then there was nothing. It was this means, and then it came dark matter, and then in 2003 they said back to this. So in any way, there is something beautiful about these geometric systems. And of course, nature has shown us to be symmetric in many ways. So they've been searched in many concepts. For example, also Kepler thought that the motion of the planets was to be following some geometrical shapes of the Platonic solids. But they do exist in the nano world and in living science. So here's an example of the viruses. We know that when they start having certain larger sizes, they are spherical, but then they buckle and form an icosahedron. And also the fullerenes have these sort of large fullerenes have the shape of an icosahedron. recently, not so recently, but in the latest 10 years or so, there's been some mixtures of cationic and ionic surfactants that when they form vesicles, closed shells, they buckle into icosahedral shapes. And I'm going to talk about some of the examples that our group has been doing on a smaller length scale. These are micron size of regular or faceted shells. So these are contrary to the solid shapes that are in wolf shapes of metals. These are just shells that are, they have nothing inside or they encapsulate something, but they are shallow shells. So why were I studying these things? Well, first of all, can we design some membranes with particular shapes and distribution of matter around them? And we would like to know if we're able to make very asymmetric shapes, because in principle, if they have more lack of symmetries, they probably recognize or they can be more functional. Like you can concentrate components in the vertices and the edges of the object, as opposed to if you deliver something in a sphere that is very symmetric, it doesn't have many properties, right? It's more inert. And we would like to look at whether they are, in nature, these microcompartments that have this lack of lower symmetry structures, and what are the requirements for that. So the other more complex solids are the Archimedean solids that are like a Platonic solid truncated. So now instead of only one polygon, you have two of them, but all the vertices are equivalent still in these ones. And when you get to more complicated polyhedra that have less symmetries in some way, are the Johnson solids that now they are not all the vertices equivalent and so on. This is another example of objects studied by mathematicians. These are n-gonal icosahedra, which is like these balls that are half moons that you close. And I'm going to show you that all of these shapes arise also in elastic or crystalline shells. And so what is necessary to form these is to go to something that is made of more than one component. So if you organize things in a shell, I'm going to show you that in a closed shell, like a vesicle, but you have more than one component, naturally there's going to be a segregation of the components to form something with very low symmetries. So I hope I have persuaded you that this is a sort of like a new way of understanding heterogeneous systems that open out and face a space much more possibilities for forming. These microcompartments can act like catalytic structures in biology, like organelles, that they concentrate components that absorb specific components. So I'm going to give you first an introduction of what is buckling of elastic systems that close elastic components or crystalline systems. And then I'm going to study the modeling of what happens when they are heterogeneous, when they are not made of a single crystalline component, but more than one, and show you some experiments when we reproduce that. And this is an example of organelles, which exist in cells as catalytic structures. When you go to the textbooks, they're always drawn as an icosahedron, because that's the only shape, regular shape, that is sort of ubiquitous and people have found when you have something faceted that is crystalline. And so this is an example in the textbook, but when they have trying to reconstruct them, here you can see that actually they have shapes that are not really an icosahedron. You cannot make projections and find these shapes. So basically it's why, first of all, I'm going to explain why the icosahedra is so ubiquitous, and then what do you need to find other shapes and what are the properties of them. So these are very important because these are, in these organelles, they are called carboxysomes, these microcompartments. These are found in bacteria that fix carbon dioxide. So these systems are capable of absorbing particular type of ions and make energy, other components and so on in the cells. I also want to go and show you that there are other structures that also show these sort of faceted shells, and these are found in archaea organisms or halophilic organisms. Archaea organisms, they used to be believed to be bacteria too, but there are different types. Instead of having a bilayer that is a cell that covers the cell, like in bacteria, they are made with surfactants or lipids that are actually connected to polar groups and one tail only. Okay, and what you can see here is that these ones are faceted, but they live in a very high concentration of ions and divalent ions and so on. And when you chelate the divalent ions, these structures become spherical. So basically, this is a hint to tell you that electrostatics is causing a lot of the inducing the crystallinity of these objects. And why do they need to be crystalline? These are square ones. Why do they have to be crystalline? Is because they live really in conditions like brine conditions. So there's a lot of salt around them, and they have to be very robust to be able to survive. Bacteria can absorb a lot of salt as opposed to the cells of humans or other animals, more complicated. But still, these systems can actually live in or survive in conditions of very strong salt. So they are crystalline shells, the surface. And the other thing that had been puzzling biologists is why they are square, and they didn't understand where the square shape came from. They have done analysis of X-ray, and they knew that these structures are made of hexagonal packing triangles. So they were not really made from hexagonal lattice. Nevertheless, they buckle or they had shapes of squares and that didn't follow the symmetry of the crystallinity of the unit cell. So these are the mixtures of the experiments I will show. That I will try to make some of these shells. So using electrostatics, we pick up a lipid that has the possibility of ionize at 3 points. So it has up to 3 plus charges, and we're going to mix it with one that has -1 charge. So this material is not water soluble, so it will be a salt in water because it doesn't have enough, degree of, it cannot ionize to really solubilize in water. And these ones for myself, that are spherical, which they show the charge to the water. But if you mix them, you make these sort of very low structures or shells. And I'm going to show you simulations and some experiments. And what we find is that indeed, we can have very low symmetry shell structures, but only when the packing of the molecules is hexagonal. Okay? So that was in agreement with what it was known before for these structures, that they have low symmetry, but it's a hexagon. And I'm going to try to explain the physics of why you can only have these closed-shelled shells with certain crystallinities, symmetries in the structure. So let me just review what are elastic versus fluid membranes. So you probably know a lot of the lipid membranes that are fluid. They don't have a Young modulus, right? And as soon as you have a crystallinity in the surface, so that they can have a Young modulus of stretching, then these shells can have various different physics. One is that they buckle, and I'll show you why they have to buckle when they are large. I mean, it's very natural. They cannot continue being spherical, like a vesicle. Or when they go to very large sizes, they also crumple, right? Like a piece of paper like that. If you take a little different pressure from inside and outside, they can crumple the surface or fracture. So I will concentrate in this smaller one that buckle. And so how do you describe something, a membrane? There are very simple physics that people use to describe membranes. And this is the simplest Hamiltonian, which you say, well, the membrane has H0 is a spontaneous curvature. So the membrane wants to be flat, H0 is 0, or if it has a normal bending to it, it has a different from 0 bending. So you pay a penalty, quadratic, in how much you deviate the mean curvature at some point in the membrane with respect to the spontaneous one it wants to have. And the coefficient here is called the bending rigidity. So even if it's a fluid membrane, it has a bending rigidity, okay? And that depends on how is it made, what are the molecules that the membrane is made of. And then there is another term that people call the Gaussian rigidity. And this is proportional to how much Gaussian curvature you have, which is the product of two main radius, and the mean curvature is the sum of the inverse. And so any point can be characterized from two radiuses, R1 and R2. And so these are the energies. So everything depends on the integral over the sphere or the surface, if you close the membrane, and that gives you the bending energy of the membrane. And then there is another part, which is elastic. Then you have to add the strain tensor, and we're going to use a work of Song and Nelson that had been able to discretize this very complicated continuum elastic model And we're going to use it for a closed membrane to show you how we model these membranes that now have a bending and elastic or strain energy. So the strain energy is again quadratic, but these coefficients are related in the model of Song and Nelson to the Young modulus by some multiplicative factor. So the physics of how something like that, a closed shell, elastic shell, buckle, is very simple to explain without having to go to the math, even though if you do the discretization of that model, you'll get the same. And it comes from the fact that if you have the most closed packed in two-dimensional structure, which is a hexagonal, and you close something with a hexagonal pattern, you cannot, right? You will have to, like if you wrap a ball, it has to spike. Because there is a theorem that it's impossible, basically, to have a crystalline structure that is closed without defects. So defects are, in this case, are points where instead of having 6 nearest neighbors, you're going to have 5 pentagons. You can have defects that are heptagons, that have 7 nearest neighbors, or 5. But there are very strong theorems to tell you where are the ones that cause lower energy, okay? So the ones that cause lower energy are the pentagons. And it comes like that. The number of points of the crystal in the sphere that have 5 nearest neighbors minus the total number of points that have 7 have to be equal 12, 1 minus g, where g is called the genus of the structure. The genus, the topology of a sphere, the genus is 0. So it has to be equal to 12, no matter what. Okay, so if you think of it, so you can have these are the genus 0, 1 handle is the number of handles. So if you have a genus 0, you need to have, you look at it, the minimum is to have 12 pentagons. Because if you have 13, you need to add 1 heptagon, right? So what happened in physics is that this minimum number are what we call the on-screen defects, because they hate each other. Every time you put a defect around of 5 neighbors, if it doesn't have close to it an heptagon with 7, it will grow to have a strain that will grow. So it has a lot of strain. So what it means, this theorem, which is actually a mathematical theorem, means that everything that is closed, the lowest number of defects you need is 12 on-screen defects that are pentagons that will hate each other. So if they hate each other because they cause a strain, then doesn't matter what is the shape, it has 12. It can be distorted or whatever. But in the sphere, if there are 12 of them, where do you think they go? Like a donut, you don't have to have defects because the genus is 0. So you have 0 defects. You can always wrap a tube and close it in a donut, and it's very perfect. But here, you have to have those. So where do they go? They go inside what is an inscribed icosahedron. If you draw, the icosahedron has 12 vertices. So, and they're further away from each other, right? So if you have something periodic, and they are in the 12 corners of the icosahedron, they are equally spaced and further away from each other, and that's the minimal energy of something that has to have this effect. So anything that is spherical already has icosahedral symmetry in that sense. And if you add other defect, or pentagon, it comes together with the heptagon, they come together and they form like a dipole interaction. They come close and they get rid of the strain at longer range. But the 12 ones, you don't get rid of them, so you have to, if they keep growing, if you have one defect, say in a flat thing that has a pentagon here, and it wants to be 6-fold, and you keep growing it, the energy grows quadratic with R squared. So eventually, it cannot continue the energy of having this defect in a flat crystal gets so large that it has to buckle up to release energy. So here's the buckling transition, it goes up. And so this is exactly the buckling transition that you have if you take something, a circular thing, and you divide it into 6 pieces, and you remove a 6. And then you want to close that. It will form a little cone. And so if you have 12 of these defects in the sphere, and you get very large viruses, the radius of the sphere keeps growing, it has to release the strain by buckling, but the 12 of them buckle simultaneously. So the shape you end with is an icosahedron. And that's what is so ubiquitous. So you have something made of something, one crystallinity, one type of crystal structure, that wants to have hexagonal closed pack, eventually will buckle into an icosahedron. And so that we know it, and well, it was explained by Nelson, even though for a many long time, Caspar and Klug and people that study viruses knew about the icosahedral symmetry, but it didn't mean that they knew it was faceted. They only knew that a sphere had icosahedral symmetry. It was much later on the physicists have explained the buckling of it into an icosahedron. That was in 2003 only. And so people have now shown it and so on. So what happens if you have, instead of one type of component, you have two components that are making the shell? So you have to generalize this Hamiltonian that I showed you before that we use for the bending part. Now you have this coefficient here that depends on the position in the sphere that has two coordinates, right? Because it's a plane, the sphere, so you have to define two coordinates as 1 as 2 and integrate. So this becomes more complicated because now, this is not a constant that you can take out of the integral or whatever. It's a function of position, and when you integrate over all the sphere, counts differently. And so we have the same terms, but this is a function of position. And so now all of the theories that people have used before for something close are more complicated. Why? For example, if this was a constant, the Gaussian curvature, it will come out of the integral, and there is a theorem that if you integrate the Gaussian curvature, it's equal to 2 pi the Euler characteristic of the structure, which for a sphere is 2. So it's always 4 pi. But now if you have a function of r, you cannot take it out, so you have to take it into account. And if you want to make this for a heterogeneous system, this theorem changed now because it can develop what is called geodesics, lines or curves in a curve. These are called geodesics. So what is happening when you have two components is that you can develop actually lines in these surfaces that I'm going to show you that is what causes the buckling of it, because it's much easier to buckle something making these lines, minimize the energy, than actually buckling where the defects are. And this is what I'm going to show you, okay? So I'm sure you have heard the term geodesics, like a geodesic. So these are, if it was a spherical and you put a curve in a curve that doesn't have the same curvature of the sphere, it will have a contribution in here, the geodesic curvature. So that will minimize the energy. And then you have also worse. The elastic part, it becomes even more complicated if it's not homogeneous, because these coefficients are functions, again, of the position. So the way we solve this is numerically. So you discretize, following the Song and Nelson discretization of this continuum, we discretize the structure. So what you do is you tile, a sphere with hexagons and the 12 pentagons. It's fixed the connectivity because it's elastic, so it's a solid. And then you allow to have normal vectors in these triangles, and you can pull them out and change locally the angle between them and change an energy proportional to this coefficient. That's what we do numerically. We pick up a point, a triangle, and we poke it out, get it out, and look at the angle with the neighbors and see how much the bending rigidity change. And then you have an elastic part. An elastic part is made of springs that have a normal characteristic L0, and you stretch them when you poke these things out. You change the spring, and you calculate the energy with this coefficient. And these coefficients, in the parametrization of Nelson, are related, this to the bending rigidity of the Hamiltonian I showed, and this spring constant to the Young modulus of the membrane. So these are mechanical parameters. And this model has also the Gaussian rigidity is equal to minus twice the bending rigidity. So it has both terms in the Hamiltonian that I wanted to model. It has these terms and it has this other term. So by doing this, we pick up systems. I'm going to calculate what patterns I have if I put a fraction of two components that have different coefficients, K's and spring constants and Young modulus in the two systems. So I'm going to solve first the case where they are chemically compatible. So I'm going to choose two components that love to be together so I can coassemble them in a single shell, mechanical shell. And I'm going to follow and show you that even that case will show to segregation into these lines where it will buckle to release the strain. And the patterns are coupled to the shape, and we self-consistently determine the shape by doing this sort of numerical minimization. So here's the case of, we're going to pick up one that is rigid, that still hasn't buckled, and mix it with one that is already component that will be buckled for the same size. Okay? So one is more easy to bend than the other one. And when you mix these two, even when you just mix them here together in a flat, you start having some patterns interesting. Where does this soft one concentrate? When they're more easy to bend? But in the sphere, this is sort of like a gallery, an example of the shapes that is in the poster that they show. This is 100% the one that is already buckled. And so the only shape this component can have is an icosahedra. And this is the case in the opposite extreme, it's 100% the rigid one. So if you start adding into this rigid one that is a sphere still, a little bit of a flexible material, the first thing you notice is some very interesting theorems of physics, which is that they form lines that are vertices with 3 legs. You know, it's like in the sub-bubble problem, you always have in 2 dimensions trying to form these 3 legs. But then these 3 legs are of the softer material. So the softer material, we just spot the component, blue, and it aggregates to form these lines. And as soon as you have a critical amount of the blue that it can join the lines, it buckles, and it forms very low symmetry shapes. So it bends where it does it to be able to bend and release the strain that is happening because it's a crystalline in a sphere. So you go through all of these shapes, you know, up to you concentrate more and more the flexible, and you recover the only shape. So the point is that if you have something homogeneous, you have 2 possible shapes for anything that could be closed, shell, and in the moment you have 2, you have a family of possible shapes. And so you can form the actual magic, any of the possible Platonic solids, and the ones hosahedrons that I told you about in the beginning, half moons and so on. So the other interesting thing is the mechanism by which this buckling occurs is the opposite of the one that Nelson have put together with his student. Here, if we calculate the mean curvature of the points on the lattice in a plane, planar plane, this is the buckle structure, and here is the easy to bend. These points that are dark is where the pentagons are, because there's still 12 pentagons in the structure, the defects. That's a theorem. They don't go away, right? They are actually sunken in. So the mean curvature is lower where the defects are, as opposed to spiking out. But the mean curvature concentrate, where it's easy to bend, is the lighter color. And that's very similar to something that Tom Whiting has done, that when you crumple a piece of paper, it doesn't inform that you concentrate the mean curvature. So here, you know, the buckling follows that. And so, you know, we can make the patterns of this mean curvature along, and definitely, you know, it's concentrated along the folds and so on. So these components depend on the fraction. I told you the shapes depend on the fraction of the flexible one in the structure. And if you start adding a degree of incompatibility, suppose you change the media so that now the blue and the red don't like each other. You can form other Janus-type particles when it's segregated. So in that case, if the red likes to be with the red and the blue with the blue, and they pay a penalty if they are together, then of course they would like to have the lowest interface in the structure. So they have different curvatures, mean curvatures, so they form different curvatures when they close. But nevertheless, if you calculate here the magnitude of this energy versus the fraction of the soft component, here's very little, you have to be above certain amount of degree of incompatibility to actually coarsen them and make them come together, the lines. So you can generate very large number of shapes just by making shells of 2 components. You can imagine if you do 3 or 4. So how do we make these shells? And so we use the surfactants I told you in the beginning. And it's lipids, you know, or surfactants that when they are charged, they can form micelles or vesicles that are called also liposomes or flat membranes. And so when we are able to coassemble, if we coassemble 2 that have opposite charge, the driving force to them to coassemble is electrostatic, because this minus will attract, these plus groups will attract the negative. So even if this one doesn't form, when you have a surfactant that has one tail, it forms micelles. If you have surfactant with 2 or 3 tails, they start forming bilayers. But when you mix them, you can show, we find that they form bilayers. And here's some TM microscopy where you go and you have the cation alone, and you can see the micelles. And here is when you mix a cation and an ion mixture, that you see these sort of shapes that are buccal shapes. So this is a quick freeze deep edge TM. So let me explain you why will they form crystalline structures? I mean, if it's buccal, it's crystalline, right, as opposed to sphere. So why do they crystallize when I mix 2 surfactants? And basically, it's driven, the aggregation of them together to form the bilayers is driven by electrostatics. And electrostatics brings the pluses and minuses together, and you can sort of estimate how much energy you gain from co-assembling them, right? Basically, if you were in a solid, the ionic bonding, if you put 2 charges, plus and minus, in the electric permittivity epsilon 1, at D around 5 angstroms, this thing is 100 kT. I'm sorry, the energy is 100 kT, right? So it's very strong, it's ionic bonding. There is a precoefficient called the Madelung constant from adding plus, minus, plus, minus. If you put them in water, and you have them at the same separation, because this electric constant is 80, so basically the energy is of the order of kT. So salts in water that don't have hydrophobic groups, they always dissolve unless you concentrate them a lot, okay? There is one length scale, which is called the Jerome length, at which the separation between 2 particles in a given media is equal, energy at that separation is equal to kT. So if you add, in water, this LB is 7 angstroms. In a solid, it's 100 angstroms. And what it means is that if you have 2 charges in water as close as 7 angstroms, they bind. But they are further away than 7 angstroms, they prefer to be free, kT, entropy, okay? And so in a metal, you don't, when you have the electric constant 1, you don't have excess charge alone, because even 100 angstroms, they will know to pair. So you always have defects, like in ionic crystal, you will have defects that keep a stoichiometric locally. But what happened in this surfactant is that mixtures is that they are only in contact with water, the head groups. The tails are in a lowered electric medium. So this length scale at which 2 things correlate, or they know they have to form pairs or be together, it changes. Instead of being 7 angstroms, it turns to be like 15 angstroms for that same separation, okay? The Jerome length. Because the interaction between these head groups that have charges is weighted by the mean dielectric constant. So in water it's 80 and here 1, the mean dielectric constant is 40. So LB, that distance that was 7 angstroms, is double. It's like 15 angstroms. So if you calculate how much energy we'll have seen from the top a membrane that has positive groups with negative, making electron neutral, then this energy, adding 3 times these pairs, by pairing only, it's like 10 kT. That's not enough to be a solid, right? Still for this. But what happened is, and I'm going to show you, is that when the D is around 5 angstroms that we get from the X-ray, the tails are close together because of the electrostatics, and so they crystallize. And when you crystallize the tails, for each carbon here, you get 1 kT. So it's units that have 4 tails of the order of 10 to 12 carbons has, when you add the electrostatic to that, you get an order of 50 kT, and that's a perfect solid. And that's why we actually be able to crystallize the structures when we mix them, these 2 components. Okay, so that's sort of like the explanation. And here is when you go to the small angle X-ray, this is the work of Betzig in the group, you can extract the width of the bilayer. This slope tells you that it's a bilayer. And then you can look at the wide angle and look at what are the symmetries, what do we see, we compare to the TMs, and see what shapes do we see. And we do this analysis for different pH values. The pH values change the degree of ionization of the head groups. So we can change the degree of charge in the membrane by changing the pH. So what we find is some reentrant, like pH 3 to 7, we find closed vesicles. Then we go to a pH 7 and 9, and we have ribbons or sheets broken, so the vesicles break. And then if you go to pH 10, you again have closed end vesicles. So this is a puzzle that we find, and so we try to look at the wide angle and see what was the symmetries at the different pHs. So what we find is that at pH 3 and to 7, they were hexagonal, one peak for the tails, packing. And if you go to pH around in the middle, like 7 to 9, they were very low symmetry structures. It was a rectangular seam. And so basically you can see that when you coassemble two structures, if you calculate the degree of dissociation of the groups in the water, how much charge it has each group, if it was by itself, it has a curve. Here's the pH. The blue, for example, is the one that is positively charged. It's charged, and then at pH around 10, it gets zero charge. And the red is the negatively charged. By itself, at pH 7 or so, is zero charge. But when you have them coassembled, this opens up. So you can have the cationic, the anionic structure being charged up to when you have, say, at pH 3, 30% of the group is charged if they are coassembled. So we do a simulation with different fractions of charge to simulate the pH and calculate, how they are correlated, the plus and minus at those pH values. And from those results, we do full atom, where classically we mix, in this case, the one that is green, the one that is, what is this color? Gray, is non-charged. The red is negatively charged, and the blue is the one with the 3 positively charged at 100% charge, because this is at pH 3. And so we match and look at what happened compared to the X-ray values for the thickness. And we do it for the pH 8 also, when 95% of the groups, the negatively charged groups are charged. So what you see here is that when you charge more, both of them, the bilayer collapses. Okay? So it changes a lot the thickness. And so this collapse, and it matches the X-ray. We believe it's because these head groups that are charged, these are neutral, they're neutralized. They fill up the space in the bilayer hexagonal, but when they all charge, they come closer to the head groups, and so you have to fill up that space when the floppy big side groups of the 3+ come down, and it collapses the bilayer. So when it collapses the bilayer, it changes it, instead of being hexagonal, it becomes rectangular. And if we go and increase the pH from 3 to 8 to 9.5 and higher, it goes back to be hexagonal. So we have this reentrant transition from hexagonal, rectangular to hexagonal. And here is when you have a hexagonal, the tails are free to rotate, right? The symmetry of the tails. And if you were to have this rectangular, it's like if you have deformed these cylinders, and they don't have so much degrees of freedom, so they have lower symmetry, the structure. So we have this reentrant transition where the degree of dissociation goes from pH, you go from triangular lattice to rectangular to triangular, and then here is closed vesicles, then open vesicles, and then closed vesicles. So basically the model that the previous simulation was a full atom with all the water that I didn't show. This is a coarse grain model Martini to be able to look at larger assemblies of the particles. And when we put 30% neutral on the negatively charged and the positively charged, and you take a little bit of water, this is the shape it takes. So you can analyze here the order. This is hexagonal order. But where it bends, it cannot really be crystalline because it's bending the structure to close. And so one of the things we know you can do many models to explain this. When we do the continuum, we can also mix some crystalline, more rigid, with some more flexible, and get related shapes. and either following the model I showed before or doing some continuum mechanics model of thickness. If this thickness is different than that, it's like two components with different mechanical properties. So basically, having these mixtures of plus and minus allows you to change the actual mechanical properties at different points because they have different degrees of charge and different degrees of flexibility. So it's like the model that I showed in the beginning of two components, mechanically component systems, matches, like in this case, by changing the degree of charge because you concentrate materials in different points and they have different thickness, so they have different mechanics. Okay? Then the other thing to notice is that these are able to close because if you have a system where you have disordered tails in these regions that are very deformed, the number of nearest neighbors it wants to have is 6, and the tails wants to have the rotation of the hexagonal. So these two components, mechanical parts, this one, coexist. And that is known, for example, in monolayers. If you have a monolayer crystalline in a hexagonal, it can coexist close to it with a disorder. But if you were to do the same, and these are the shapes that we find in TM, if you were to do, let me see here, in this case at pH 8, it's not a hexagonal, it's a rectangular. So it is impossible to have something with so low symmetry, rectangular, coexisting with something that has to be disordered to close. So they cannot support in this lower symmetry tail structure something close. So that's why this sort of like, even if you have very low symmetry shapes like these ones, the unit cell is hexagonal, like in agreement with they found in these, archaea and other structures studies. Now, the thing that we find is from the X-ray, you can also try to measure the size of the domains that are crystalline, which is of the order of 25 by 25 nanometers square, extracting from the width of the peak. And so when you try to do, if you have this size, and you think that these plaques of crystalline come together, if you have something that is going to close in a small, it's going to have very low symmetry. Now, if you try to bring these things in a surface much larger, it's going to look spherical, right? So contrary to the other buckling mechanism, in this case, we find that the very large ones become spherical, and only the very small ones have very low symmetry. So it also matches with the X-ray size. If you were to put them into these 100 nanometer, these sort of structures, it will have to be faceted. But if you take one that is much larger, it will look like this, spherical. Okay? So these two things explain that. So now we went and tried to look what happens, say, if you put 2 to 1. So what is the effect of the competition of electrostatics versus van der Waals of the tail's crystallinity in forming these transitions that are reentrant? So can you drive this in other systems? In biology, everything around 6, 10 kTs energies is very important. And they all, the hydrogen bonding, the kTs, everything is very of the same order, and they get correlated on order of 10, 15 kTs structures. And they're stable, and some are not. So when are we able to change the structural change in pH if we change the length of the tail, say? And in that case, we're changing the balance of electrostatics versus van der Waals, right? Because the tails want certain symmetry. I know, for example, that the cationic lipid that doesn't mix, the anionic ones that don't mix in water, they are always rectangular sea salts. So that's the lowest energy you want to have. Nevertheless, by coassemble them with another 3+, we were able to do hexagonal. So, there is a play of energies here. So we were able now to coassemble 2 pluses with -1 of different tail lengths. And we found that, what are the symmetries as a function of the length of the tail. Here is carbon, has 14 carbon, 16, and so on, of both of them. We changed the length of both of them. We were able to access the surface area and the symmetry that the structure has. So you can see that, the hexagonal suddenly for this one is another symmetry. So there is a transition. Went from hexagonal to this, I think, is rectangular C structure. So this is the structure it has, the pure anionic component, always. So basically here we show that only when they are in a balance of the order of 16 carbons, 18, up here you have reentrant transitions with pH. So if the tails are not very long, you're able to change the symmetry they want to have if they were solid to hexagonal, and can go from hexagonal, depending on how much charge fraction of them have, to rectangular C. Then we have another phase, new phase, rectangular P, rectangular C, and so on. So if we went to less electrostatics, we were able to actually open more symmetries. So what I'm trying to show you is how, with just one single type system of tails with head of charge, that the charge can get ionized at different fraction, you're able to go through cover a phase space much more broad. But it only has a certain range of where these things work. And this has to be in a range where energies are, of the order, you don't have something that if it's very strongly biased for the tails, like here, 22 carbons, it wins over all electrostatics. So that just makes it like inert more. And if you are in a range where they are more competitive, 10, I remember I told you it was like 10 kTs for the other one, electrostatics versus 40, then you can access more, a broader range of structures and reentrant transitions and so on. So I'm just going to now summarize here. These structures that are in a hexagonal can be closed, can have genus 0, and have the defects that the theorems hold for structures that are crystalline closed. And when you have structures that do not have hexagonal symmetry, they don't support a closed structure. Okay? And we have these possible transitions and a minimum value of co-assembly when the tails are too short. And why the square lattices will not close? I try to explain you that because there is no possibility of coexist of something that has 6-fold nearest neighbors in a disorder with something that has symmetry of 4, a square or some other lower symmetry, but has to have the same symmetries when it folds. And I have here an example. Suppose I have an ionic crystal of plus and minus charges that wants to form a square lattice, right? It cannot form a triangular. You need to have a 2-to-1 or 3-to-1 to form a triangular. So if you make they're in a droplet, and then I'm sucking out material from the droplet so that the curvature, they're concentrating more. So it comes a point where you actually change the symmetry. And it's not that they cross back and become triangular. So basically, structures, a square or lower symmetry, do not support a very strong curvature, and so on. So I'm just going to summarize that we find these regular polyhedra in the structures, and that they spontaneously seem to buckle, and they have, the large ones have icosahedral symmetry, but when there are two components, they seem to be forming more lower symmetry structures that supposed to be more functional because they're like decorated, with shapes and compositions. You concentrate the concentrations of different components. And this closed shell seems to be having always hexagonal symmetry. And the subtle balance between them allows transitions that can be monitored by changing pH, but only when the energies are of related order. So I'm going to, these are some of the experiments. I show experiments that also seem to corroborate these statements. And this is for modeling, what do we need is this appropriate coarse graining with minimal chemical details will allow you to get a lot of the physics. And there are more important effects we have to take into account with mean fields. So the electric heterogeneities from water to oil to explain the things, and elastic interactions. And we were able to cross morphologies from the full atom to the continuum models and be able to study the structures. This is just a diagram of all what I showed to summarize the X-rays. And this is the full atom, this is the continuum, and this is the coarse grain. So we were quite pleased that they sort of match the physics well. And these are the shapes. Thank you.
Questions for Monica? Let me actually start off with one then. So do you get any qualitative differences, for instance, if you move to a three-component system? Is there new if you move to a three-component system, if you move to more components, do you see new phenomena?
I bet they can have more complicated shapes, yes. I think that, basically they're going to have the same principles. Okay. They are going to segregate to induce the buckling, and they're probably going to do it by the minimum path first. Okay. And so now if you start having incompatibilities and so on, maybe they form more complicated ones. But the first principle is, yes, that it's, if you are not paying a lot of energy from interface, in the first principle, you don't mix things that hate each other.
Right.
But the point of view is that even if you mix them because they would love to be together, they will segregate to the least strain. Okay. So elastic energies are non-local energies. Right. So they're very strong, and they govern the whole shape of the object.
But they do seem to localize as well, in the sense they localize as well. You get segregation.
You get segregation just because of this non-local force that wants to be minimized, elasticity. Yeah.
Shout, it's just as good.
You have an amazing array of different polyhedral shapes, starting from the spherical down to the atomic. And I guess you can change them by location. But do you have any insight as to how it actually changes the mechanism by which it goes? It's sort of this equilibrium.
Right. Right, it might be that some of these things, who knows, is really the lowest energy state because they are finite systems. They are not phase transitions like, in infinite systems. So you have that depends on R, how many I put, points. So it's a discretized model of a continuum that I try to make it like a binary alloy type. And, basically, I don't know the path because this is just the lowest energy state, 0 temperature. So you do it with simulations going to the lowest energy state, annealing. There could be barriers between them. I'm just starting with the spots, and then you minimize, and you keep lowering the temperature to get the one that doesn't change. And then you start with another initial conformation. You don't get exactly the same shape, but you get very similar shapes. So the mechanism of saying these things want to go to a structure where they can release it by bending, it seems to be very reasonable. So if you have something that can liquefy it a little bit, it will do that. Now, some viruses are very strongly crystalline, and the only way they are very homogeneous, and they just buckle into icosahedra. There is no segregation of anything. But if they are able to have a component that liquefies a little bit more the structure, then they will not form the icosahedra. They will form these other lower shapes. Like it happens in these archaea. They have other ways, because they are multi-component. The archaea have... Yes, if you have something that liquefies, will destroy the virus or whatever. But from other shells, it might be more important, other properties. Like the capsids have the duty to protect the virus, the RNA or DNA inside. But the carboxysomes want to attract certain ions in and not others. And maybe the vertices are very important because it's more soft there and they can penetrate and have different permeability than the other parts. So I'm not saying that is how the biology, but I find a way that you can actually form other shapes. When I found them, I didn't know they exist. And I asked a biologist, I said, I think they are ubiquitous in biology. And he told me, good luck publishing your work, because no one has seen them. So I went to the literature, and they called them quasi-icosahedral. And they had just found them around that time in 2008 and so on. Yeah.
Paul?
In biological systems, the membrane chain length is a typical length. I think it's 16 or 18 or something like that, which is similar to the length that you're seeing these effects. So is there a biological function to
I don't know exactly, but it happens to be around, I will think things that are 15 kT, 10 kTs playing with each other range. It is working, that they can change and be open up and reversible. So other people tell me they found these energies too in biological system. I'm not going to make, statements bigger than that, but in this case, I know that when we went to 22 carbon, that's it. They're just one structure.
Lynn?
Right. The systems, like for example, you will be talking about will be like if you have oil and water, and then you have surfactants that go there, then it does depend how many absorbs with chemical potentials. And then, the charge of them, and they can crystallize too if they're flatter or less curved, or they might buckle, these sort of emotions at some point. Like if you have conterions hard that in the tails, and they want to be crystalline, you might buckle something that's supposed to be an emulsion if you crystallize the surface. In that case, the chemical potential matters. We don't have chemical potential here because it's already totally crystalline in the shell. I just minimize that shape. So I already assume that it has been cross-linked, the tails, or that in my mechanics model, okay? So I didn't have an extra component. Like if I was doing them for a monolayer at an oil water, then if I deform, every time you deform a shell, you change the volume. You can change it doing the volume of the area, right? What do you keep constant? So in the case of emulsions of oil and water, you will have to keep constant the volume. So when you deform it, you stretch it, and you get more area. So then the chemical potential of how much more surfactants come in matters to keep the other energies, the interfacial energy constant, not increasing a lot, and so on. I'm trying to do that case. It's much more complicated. But it is interesting, yes. You have to put the constraints, and then what is balancing the extra forces and so on with that chemical potential that you mentioned, how much it absorbs to compensate for other energies that are increasing when you deform the shape. Does it make sense? Yeah. Okay. So. But not in this one. This one was just fixed number of particles, and I let it stretch. They changed the area and the volume accordingly with just the stretching and bending. I don't have any other surface energy term increasing if I increase the area or the volume was able to expand. I didn't put a constraint of constant volume in that.
Oh, right here, Michael.
Right. I would love to see whether, for example, if you label something, if you can see that there is different concentration in different areas. For my model to be correct, I will have to have different percentages of, say, the positive and negatives in the corners and the flat. I don't know enough of the techniques to know how to do it. I know maybe neutron scattering, putting some tails of the cationic with the alterated and not the other ones or something like that. You might be able to look at the length scales of they are segregated or not. But, these things, the TMs were done by, in the group of Sam Stoop and Michael Betsick helping with the students doing these things, and I'm not so familiar with the technique to look at it. I'm looking forward to someone to see, if you can actually image things that have segregation of components and you can tell where they are. And then, according to the properties, to know why they went to certain vertices or corners. Or, I don't think this is known yet.
Okay, let's thank Monica again for a very nice seminar. Thank you.